These teams to find a resolution for the problem situation. What's a metabolic disease? These are a group of diseases which are inherited, and its main characteristic is to disrupt the normal process of converting food into energy within the cell. This type of diseases affect the functionality of the cell to execute important biochemical reactions which involve the transport or processing of biomolecules. Metabolic disease we're focusing on is Gaucher disease, which is caused by the mutations in the GBA gene, which results in the lack of an enzyme called beta-glucocerebrosidase, and this causes the body to be unable to break down lipids.
Now this image shows the metabolic pathway involved, sphingolipid metabolism, where hydrolysis of lactosylceramide occurs, and the enzymes involved are beta-galactosidase and beta-glucocerebrosidase. These are the reactions that involve the previously mentioned enzymes, and as you can see, the product of one is the reagent of another, and here the sphingolipid breaks down by the glucocerebrosidase.
Enzyme replacement therapy is a type of therapy which can be performed with the usage of a recombinant enzyme that performs a specific function, in this case, break down glucocerebroside. The sources to obtain the enzyme needed can vary, but some are Chinese hamster ovary cells for imiglucerase, fibroblasts for beta-glucerase alpha, and card plant root cells for teleglucerase alpha. Obtaining the desired enzyme can take a series of complicated steps. Gene insertion in host organism, harvesting the organism, and extraction of the enzyme, through sonication or homogenization, purification through a series of chromatography, and finally a quality control based on potency and purity. Potency in this case being the ability to break down glucocerebroside, and purity being the amount of impurities in the enzyme.
Characterization allows for determining their activity, stability, specificity, and kinetics, also knowing heterogeneity, the impurity profile, and the potency. So our recombinant enzyme must be characterized to ensure its safety and efficacy. Bmax is a parameter that will help us determine the efficacy of the biotherapeutic, and also Kcat and Km are required properties for the desirable enzymatic activity.
Some of the specifications that a biotherapeutic must meet according to the FDA are high purity. It should be at least 98% pure to be safe. Also, the molecular heterogeneity in terms of size, number of individual polypeptide chains, hydrophobicity and charge, and sometimes the degree of cellulation.
The immunogenicity of a biotherapeutic should always be investigated before the authorization of the product because it can trigger unwanted immune responses. Specifications for glycosylation depend critically on the glycoform and their degree of process control. Glycosylation is crucial for the synthesis of an active enzyme in ASN19 that controls the activity of the glucocerebrosidase. Specificity of identity is based on biological properties. In terms of dose strength, the acceptance criteria is about 10%.
The social advantages of biotherapeutics we have that due to its high specificity, they tend to reduce symptoms. Now, for environmental, biotherapeutics reduce the APIs by using renewable organisms. APIs are responsible of 4.4% of worldwide greenhouse gas emissions, and due to its low toxicity and specificity, it's less likely to have adverse effects on the environment. For economic, over a long period of time, biotherapeutics can become cheaper due to advantages in tech.
Social disadvantages include the fact that patients are not able to afford them. Also, none of the available treatments right now are effective on treating Gaucher disease type 2 nor 3. Regarding the environmental impact of them, the production requires great amount of energy and water. If they are disposed in an inadequate manner, they can have adverse effects on the environment, for example, increasing the mortality of aquatic species. The economic disadvantages of them, since their production process is really complex, it tends to increase the price of them. The prices of therapies tend to be really high, and due to the rarity of this type of illnesses, private companies find it really difficult to justify
These related are 1, 3, and 10, which have to do with giving access to these types of therapies to everyone, not only the wealthy. 6 and 14, which has to do with less pollutants from water residues from these type of therapies. And 9 and 12, which have to do with improvements in tech in order to upgrade these kinds of therapies and reduce costs.
Okay, but what can we do to improve the biotherapeutics production? First, we have industrial scientists focusing on industrial ecology. This means collaboration between industries where the considered waste could be used as a resource for another company. Another way is to minimize the water usage by raising awareness of the water supply, adopt techniques with less use of water, and design technologies to optimize the water supply, recycle lab consumables, and raise awareness of greener products in the lab. Also, we have raised awareness of rare illnesses because of the limitations and the economical disadvantage and sample process. It's very difficult to elaborate the biotherapeutics, but sharing information on social media and sharing stories may be a great idea to raise awareness. Also, have invested in research on rare illnesses by organizing fundraising events and partnering with nonprofit organizations to respond and raise awareness of how biotherapeutics should be disposed in order to decrease its environmental adverse effects. And thank you. That was our presentation.